Very good evening to one and all. And on behalf of the organizing committee of this Science Promotion Initiative, jointly organized by the National Academy of Sciences India Delhi chapter and the host institution, the Dayal Upadhyaya College, University of Delhi, I would like to welcome Professor B.Rosemary Grant, an FRS, and a senior research biologist and emeritus faculty member at Department of Ecology and Evolutionary Biology, Princeton University, who has kindly agreed to deliver a talk on a very interesting topic, evolution of Darwin's finches from ecology to genomics. And this particular special public lecture is part of an year-long series which the chapter, that is the NASI chapter and Deen Dayal Upadhyaya College, have been organizing ever since the pandemic took place. So that is from the March 2020 onwards. And we are privileged and honored that several Nobel laureates and fellow of the Royal Society and fellows of various leading international organizations and professional bodies have participated in this particular webinar series, have enlightened not only the students, researchers, and faculty members from India, but from outside India also. I have with me as my co-panelist, Dr. Sudhir, who is a faculty member in Department of Zoology of Deen Dayal Upadhyaya College and has been one of the pillars for organization of such kind of programs in past also. And here I would like to welcome and to introduce Professor Grant. So with her husband, that is Professor Peter Grant, Rosemary Grant has been studying the Darwin's finches on the Galapagos Islands since 1973. The fieldwork is designed to understand the causes of an adaptive radiation. It combines analysis of the archipelago-wide patterns of evolution with detailed investigation on two islands, that is Genovesa and Dayal. By integrating ecology, behavior, and genetics, they have revealed how species are reproductively and ecologically isolated and the causes and evolutionary consequences of rare episodic hybridization. The two mechanisms of speciation have been identified, and in collaboration with the other investigators, they have estimated phylogenetic relationships among the species of finches and their relatives, identified molecular mechanisms involved in the development of beaks, and provided genomic details of evolution in contemporary as well as historical time. The research has been published in many research papers and 3 books, most recently, How and Why Species Multiply, that was in the year 2008, and 40 Years of Evolution in the year 2014. Rosemary Grant is Senior Research Scholar Emeritus in the Department of Ecology and Evolutionary Biology at Princeton University and received her training at Edinburgh University and Uppsala University and taught at Princeton University also. With these words, I now invite Professor Grant to kindly share her screen to deliver her talk.
Well, thank you very much for that very kind introduction, and I wish very much I could be in India and give this talk in person, but this is the next best thing. I have put my husband's name, Peter Grant, because we've very much done this research work together, along with our graduate students, assistants, and collaborators. So, oops, oh, seems to be, so Darwin ends his first edition of The Origin of Species with his very poetic words. There is grandeur in this view of life, whilst this planet has gone cycling on according to the fixed laws of gravity from so simple a beginning. Endless forms, most beautiful and most wonderful, have been and are being evolved. And this Nassau image actually captures the uniqueness, but also the fragility of our planet and shows that biodiversity is its fundamental feature. And indeed, if we're hiking in the alpine meadows, exploring the tropical rainforests, or diving beneath the surface of the sea, we're amazed at the diversity of life, from the simplest of bacteria to the largest elephant or whale. So how and why do species multiply? How do we even go about studying the process of speciation? Well, we decided that the best thing to do is to look at a young adaptive radiation in which organisms have very recently diversified into several species from a common ancestor. And there we may see that the process of speciation is still ongoing. So where do we look for these things? Well, there's amazing radiations, over 600 species of cichlid fishes in Lake Malawi in Africa. If we go to the Caribbean, we can see the radiation of the anolis lizards. For every one you see here, there are 7 more species, each flapping their different colored dewlaps and behaving in slightly different ways. Then if you go to Hawaii, there are the Hawaiian honey creepers with their different colors, different sizes, different beak shapes, all have evolved from a common ancestor. This would have been a perfect radiation, but unfortunately, most of these species have died as a result of human interference. We decided to go to the Galapagos Islands and look at the Darwin's finches on the Galapagos Islands. It's an extremely young radiation. We now know that it has all occurred within 1 million years. It occurs in a pristine environment. Many of the islands in the archipelago where they occur have never had any human habitation. So any measurements we make, we know are under natural conditions and not induced by humans. Furthermore, it's a very dynamic environment. It has always been a dynamic environment. And the other thing, that islands differ very much ecologically from each other. So the radiation has all happened, as I said, within 1 million years. This is a radiation produced from full genome analysis. And the idea was that a species of tiaras came over from the mainland. These are seed-eating finches, arrived on the Galapagos about 1 million years, and from then from then on, they've diversified rapidly. And as you can see from this diagram, that much of the diversification, excuse me, has even occurred in the last 250,000 years. So 1 million years, excuse me, 1 million years ago, the islands looked very different. The archipelago looked a bit like this. The only island that's recognizable is this one, San Cristobal. The rest is a large island in the center, and these ones have disappeared under the surface of the sea. Today, it looks like this. All the islands are volcanic. They've arisen from a hotspot here, and the volcanoes have punctuated the NASA plate, which is drifting slowly at the rate of 59 kilometers per million years towards the South American mainland. So the islands have never been attached to the mainland. Now, within the last 1 million years, there have been 10 glacial cycles, and this has affected the islands by raising and lowering the sea level. So during the last glacial maximum, when water was locked up at the poles in ice, then the sea level was up over 120 meters lower than it is today. And the island archipelago looked something like this. As water was released from the ice caps, then the sea levels rose gradually. The islands became more separate, and this is what the situation today. Now, it is still very dynamic, but for a different reason today. Because it lies astride the equator, 600 kilometers from the nearest mainland, it is subject to the El Nino Southern Oscillation phenomenon, which means that every few years, the warm Humboldt current ceases, the waters around the Galapagos, this little black dot here, heat up, and you get torrential rain on the Galapagos. This brings a lot of plants, a lot of food for the finches, and the finches breed and multiply. And these conditions happen about once or twice every decade. Then there are other times when the Humboldt current never ceases, and it continues to wash cold water around the Galapagos archipelago. No rain falls, and there are droughts. And a drought can last for a year or 2 years or even longer. At this time, no rain falls, and the birds starve and the birds die. So this, we have visited all the islands in the archipelago. We have done small studies on every island, but we have done 2 in-depth studies, one on the island of Genovesa and one on Daphne. And it is one on Daphne that I'm going to tell you about today. So why did we choose Daphne? First of all, we knew from the person who'd gone before us, David Lack, that Daphne had large numbers of 2 species of finches. And one of these finches, Finches fortis, is exceptionally variable in body and beak size. So within this population, there are some small forms and some large forms. And the island is also small and it's low. Finches are resident and they cannot escape droughts as they do on larger islands, where they tend to go up the mountains on larger islands. And also they're much harder to follow. But here we knew that we have a chance of following every single member of the population. So the outline of my talk will be, first of all, I will look at Darwin's model of speciation, which was now called the allopatric speciation model with character displacement. And then I will look at the causes and evolutionary consequences of the very rare hybridization or episodic gene flow that occurs between some of the species. And then finally, I'm going to tell you about one of the most exciting parts of our study, which is the formation of a new lineage, which we followed from its inception for over 6 generations. So first of all, Darwin's model. So Darwin supposed that the birds arrived from the mainland and then came to an island. This is a hypothetical model, so I'm choosing San Cristobal, which we know was there 1 million years ago, actually 4 million years ago. And they would have met very different conditions, and they would have adapted to these food conditions and maybe and lack of predators. And they would have gradually evolved, in Darwin's terms, through the process of natural selection. Population would have built up, some would have flown to another island, met very different conditions, and then continued to fly on to other islands and finally come back in secondary contact. And here they would have, one would have become very different from the original one. The 2 will compete possibly for resources, and there will be further divergence between the 2. So this was his hypothetical model of speciation. Another way of looking at this is that the species from one island would have diverged, and they would have, on very different conditions, they would have continued to diverge. They'd get to the point when they're diagnosably different, they're morphologically different, they look different, behave in different ways. And then the divergence would have continued and then finally get to the point when they, even if they did try to interbreed, it wouldn't be successful for reasons of genetic incompatibility. Now we know from other genetic work that in birds, this time from being diagnosably different to time when they cannot breed for, or cannot breed successfully for genetic reasons, can be a very long time. For example, in pheasants, it could take as much as 37 million years. Now, Darwin's finches, we know, are only 1 million years, and so they're at this point where they're diagnosably different. So this is one of the birds on Daphne, the one that is very variable, GS fortis. The population size goes down to less than 100 or about 100 during droughts, and then over 1,000 individuals during the wet years. So great fluctuation in numbers. One of the first things we did when we arrived on the island is we caught a lot of these birds, we banded them, measured them, and then we watched to see what they ate. And we found that the small ones were feeding solely on small soft seeds, whereas the large ones were feeding on the very large, hard tribulus seeds and opuntia seeds, and taking a few small seeds and medium-sized seeds. And the medium ones were taking small and medium-sized seeds. So there was a range of diets according to their bill size and body size. The other species on the island at this time was a cactus finch, a cactus specialist, scandens, and its population size also varies from about 100 individuals to up to about 850 individuals. It's very much a specialist on cactus. In the dry season, it opens up the cactus fruits, takes out the seeds and feeds on them, as this one is doing. And when the Opuntia are flowering, as they do for about 5 months, it concentrates very largely on pollen and nectar from Opuntia. So Daphne is basically all slopes. There's a crater in the middle. It's like the top of a volcano peeping above the surface of the sea. This is the inner side of the crater. The green on the crater walls is cactus bushes. This is where the birds nest, and they also nest on the cactus on the outer slope. They go everywhere on the island, and so must we. So I'm going to just show you a little bit about how we work on the island and then get down to the science. So these white arrows are not painted on the rocks. It's just to show you that this is the easiest way to get onto this island. We have to take all our water. There's no water on the island. We stay there for 4 to 6 months. And we usually stay there for about 4, and our assistants stay on for a longer time. But we have to take all our water, all our food, all our equipment, and then take more off again at the end of the season. We sleep in a tent. Our laboratory and our kitchen is in a cave. We get up early in the morning. We trap the birds before the sun gets up, or just as the sun gets up, but before it strikes the nets. Take out the birds, take them to a shady cave. We band them, measure them, weigh them, take a small drop of blood for later DNA analysis. And then for the rest of the day, we're finding out who pairs with who, how many eggs they lay, how many fledglings. We record their song, their feeding, the food items they eat, and the amount of food resources on the island. So over the 40 years in Fortis, there has been quite a change. This is the beginning in 1973, and these lines are the 95% confidence limits on either side of the mean, of this case, bill depth. We actually look at 6 measurements of the finches, 3 bill measurements. Bills are very important because they're the tools that pick up the seeds. So we do weight, wing length, tarsus length, and bill depth, bill length, and bill width. And this would show the same for weight, but so over the years, there's been a change in average bill depth, and then a plummeting, especially here, in bill length. And I'm going to talk about these 2 points, the reasons why this change occurred. Was the same in both cases. It was due to a drought and large numbers of birds that died. Some survived and others died. So this is what part of the island looks like in a normal wet year. This is what it looks like in a drought. And in a drought, the birds have to scrabble underneath the rocks for any seeds or any food that they can get. And as a result, there's terrific mortality. 80 to 90% of the birds will die. So to measure evolution by natural selection, we've got to do 3 things. First, we've got to show that there must be heritable variation in the trait of interest, and we have 6 traits of interest, which I've told you about. And as a result, there must be some environmental perturbation where there's differential survival. And our environmental perturbation is a drought, when there's differential survival. And in the next generation, the offspring of the survivors must resemble their parents in the traits of interest for there to be any change from one generation to the next. If the survival was due to something like experience and age, then of course there will be no change in the next generation. So the first thing we did was to find out whether these 6 traits of interest were actually heritable. And the reason we did this is we caught large numbers of birds, we measured them, and we watched who mated with who. We took the mid-parent measurement. We banded the offspring in the nest, waited till they were adults and measured them again. And then we compared the adult offspring measurements with the parental measurements. And sure enough, we found that the large birds, in this case, it was beak length. The ones with the longest beak produced offspring with the longest beak. The ones with the shortest beaks produced offspring with the shortest beak. And the same with all our 6 measurements. And the heritabilities all came out between 0.7 and 0.9. So very high heritabilities. 0 heritability means no heritability. 1 is 100% heritability, which actually you never get. But so heritabilities of 0.7, 0.8 are extremely high. So that was good. They were highly heritable. So we didn't have to wait long until we got the first drought in 1977. In this drought, 80% of the birds died. And as we measured the birds through the drought, we could see that the smaller birds were dropping out, leaving only the larger ones. And it was only the larger ones that were able to crack these large, hard tribulus seeds, which were the seeds that were remaining. The small seeds quickly went out. So the birds that survived were, on average, the really largest birds. When the rains came back again, they mated with each other, produced offspring, which were also large. And the average size of the offspring was here. So we had a heritable response, an evolutionary response to a natural selection event that we could understand the reason why. So this was very satisfactory at this point. Then the next perturbation of the environment came in 1983, when we had an El Nino year. And here we had an enormous amount of rain fell. Over 1 meter of rain fell on this little desert island. And this was the most severe El Nino event for 400 years, according to Coral Core data. So and I will just show you what it looked in just very quickly in size. This is what the island looks like in a normal dry season. And in normal wet season, then leaves come on the bursera trees, and there are a few little herbs in the foreground between the rocks. And these little herbs are the tribulus plants that produce the tribulus seeds that make the difference between survival and non-survival in the drought that I told you about. But this year, the rains continued, and they continued for 8 months. So very soon, grasses and herbs grew up, completely smothering the tribulus. Then vines grew over the cactus bushes. They even grew over the trees. Cactus bushes were hidden, making it very difficult to find nests. And even a year after, when the rains had ceased, you could still see the trees and the cactus bushes draped with dead vines. And our quadrats, when we were measuring the seeds, showed that the large tribulus seeds went out, and the small, or almost went out, and the small soft seeds just exploded. So the ecological conditions of the island had completely switched from a majority of a large seed producer to a small soft seed producer. So this was, and then what happened then is that the small birds had an advantage, and the average beak depth came down here. And it stayed small, or stayed small for the next 20 years or so. And then we got this plummeting that came down. Now, this was very interesting because this was another drought, and this drought lasted for 2.5 years. No birds bred. And what was also interesting in this drought, also there were very few small soft seeds, and there was a majority of tribulus seeds. So why on earth were the small birds surviving when they had no food? Well, the answer was this. At this time, there was this large Magnorostris on the island. they had come in the 1983 El Nino at the time when there was a large amount of food, and they built up a population, and the population was now well over 200 individuals. Magnorostris are twice the size of Fortis. They have this massive beak, and they love tribulus seeds, as you can see one eating or crunching one now. So what happened was that the Magnorostris outcompeted the large members of the Fortis population for these large hard tribulus, leaving very few birds, but these birds were small. So how actually did they survive? Well, what they did was they changed their behavior, and they followed the Magnorostris around. And as the Magnorostris cracked these large hard tribulus seeds, out flew the case, they cracked the case, out flew a lot of little seeds, and they ran up and picked them up. So they were following them around doing this. And I will show you a little video of this. So here's the Magnorostris cracking the seed, little bird, Fortis comes in, picks it up and takes them. See it again. So the little birds were surviving, doing this. There were very few of them, but they bred with each other when the rains came back 2.5 years later and produced small offspring. And today the offspring are still small. I stopped this in 2012, where we stopped our really being on the island for large months of the time. But we went back to the island, and they are still, measured them, and they're still small 2 years ago. So this was exactly as Darwin had envisaged. It was a character displacement event. It took us, 30 years to find it, but, and we weren't actually looking for it. We didn't think we would, but this was just as Darwin had described, a character displacement event, which caused a further divergence of the population. So I have told you about phenotypic variation under the general environment and social environment and the fitness effect it had, but I haven't told you anything about the genotype. So were there any specific alleles that underlay these big size changes? So for this, we sent all our blood samples that we'd kept for all these years over to Leif Andersson's lab in Uppsala University. Here's Leif, and the person who did most of the work was Sangeet Lakamani, who was Leif's graduate student at this time. So what they did is they took all our blood samples and they compared the blood samples from large and small species, large and medium species, medium and small species, and they found this region that contained HMGA2. Now, this was quite exciting because they knew that it was a transcription factor facilitator that was associated with human, children, and adult heights. So mutations in this gene in humans has an effect on height. So this was it, and they also showed that the large birds had a variation at this gene, which they called HMGA2L, large, and also another one, which was HMGA2 small. So what we did is we took blood samples of Fortis, and 70 of them where we knew the measurements. They did not know the measurements in Uppsala, and we sent them blind over to Uppsala, and the results came back perfectly, that homozygous large were in fact large, homozygous small was small, and the heterozygous were intermediate. So now we said, okay, let's look at the samples before and after the drought, the last drought, and to see which survived. So they did this, and of the 20 large ones, 14 died, 6 survived. Of the 19 homozygous small, 14 survived and 5 died. So this followed beautifully. Now, initially, we were very skeptical that we would find anything like this because we thought the response, especially because it was size, would be solely polygenic and involve many loci with small effects. And we wouldn't get any large effect at any large loci. But this was a transcription factor facilitator, and the shift in allele frequency at this loci explained a whacking 30% of the phenotypic shift in peak size through this natural selection event. And the selection coefficient just at this one loci was 0.59. So an enormous coefficient and a large explanation of the phenotypic shift just at this one loci. And of course, there will be other genes that will just have very small effects. So we had a natural selection with an evolutionary response. We knew it occurs when the environment changes for reasons we understood, and that other species can determine the direction of evolution, as we were able to show in this character displacement event. So now I'm going to switch slightly and look at the causes and evolutionary consequences of very rare episodic gene flow between the species. So our definition of species is the biological species concept. It is that species are a reproductively cohesive group of organisms that are identifiably distinct. That means that they hardly ever hybridize. And this applies to all these species of Darwin's finches, that they breed for 99.9% of the time with members of their own species. There's very, very rare hybridization. In fact, Black thought there was none. He said that he'd been there for months. He looked very carefully, found none. Darwin thought there were none, and other people thought there were none. And when we arrived on the Galapagos, we were convinced by other people that there was no hybridization at all. But following these birds very carefully, we did see that there was some hybridization. So why were these reproductive barriers between them rarely penetrated? So what are the reproductive barriers? How are they formed, and why are they occasionally breached? And then what are the consequences of integration? So with the birds that I've been showing you, they're all very different. There's Fortis, 17 grams, Magnorostris, 13 grams, and Scandens, 22 grams, but with a large pointed beak, and the little Fuliginosa, which I haven't talked about, it occasionally comes onto the island. They also, so they're very different, they have different measurements, they have different beak shapes, and they also have very different songs. So this is recordings of songs, Fortis songs, Magnorostris songs, Scandens song, and a Fuliginosa song. Very distinct. We tell them apart like this, but the first thing we had to know if the birds use the same cues. So we did mount and playback experiments to find out if individuals can discriminate between their own and another species. First of all, just based on morphology, based on appearance. So what we did is we took stuffed museum specimens of female Fortis and of female Scandens, which we put on the other side of the pole. We took this into a living territory owner's territory and asked, could the living territory male discriminate between a female of his own species and a female of another species, even though she was just a stuffed museum specimen? And the answer was a resounding yes. It was the height of the breeding season, and they vigorously courted a female of their own species and completely ignored the other. We did it many times with many different species, and actually on other islands too. And we also did it with controls. So clearly they can do this. Then we asked, can they discriminate purely on the basis of song in the absence of any morphological cues? We recorded song, playback Fortis song, Fortis came into the loudspeaker, Scandens ignored it, playback Scandens song, Scandens came into the loudspeaker, Fortis ignored it. And once again, we did it many times, we did it with controls. So clearly they can recognize the difference between their own species and another species on the same cues that we were using. We also know something more about song. Song is very important. Bowman did some work in the 1950s and he showed that song is learned early in life during a very short sensitive period, during 10 to 40 days after hatching. This is not unusual for some birds. It's the same sort of thing that happens with zebra finches, for example. So there's this very short sensitive period, which corresponds to the last few days when the young are in the nest to when they're out of the nest being fed by their parents. All this time, the father is singing, so it's not surprising that they learn, the males learn perfect renderings of their father's song. Females don't sing, but we know that also females learn the song. We know this from our own pairing experiments. We know it from some other work that was done in Germany, where the females, although they don't sing, they still learn the song during this short sensitive period of time. So it's not too surprising that the young males sing perfect renderings of their father's song. Once learned, the song is retained for life, and as adults they pair according to their species song they learn early in life. So how robust is this pre-mating barrier? Because after all, it is based on learning. And it is vulnerable to disruption if a young bird hears and learns the song of another species during its sensitive period. And this happens very occasionally, but it happens if the father dies and the mother is left to rear the offspring. Then the young birds in the nest, and when they're out of the nest, hear the male of the next territory, the neighboring territory. And if that bird in the neighboring territory is a bird of another species, then it will learn that species song. So we were able to test this after the colonization of the island by Magnorostris. Magnorostris is this bird twice the size of Fortis, roughly 32 grams, whereas Fortis is 17 grams, and it's very aggressive. But nevertheless, over the years, we had 8 Fortis and 2 Scandinavians that learnt and sang a Magnorostris song. So Magnorostris song is down here. This is one of the Fortis that sang a Magnorostris song, normal Fortis song here. One of the Scandinavians that sang a Magnorostris song, normal Scandinavian song here. None of them actually bred with Magnorostris. So why? The reason was that when these little birds, half the size of Magnorostris, when the breeding season came around, they opened their mouth, started to sing a Magnorostris song, and Magnorostris whipped in as if from nowhere and just beat them up. So none of them bred with a Magnorostris. So it seems that when size difference is large, the barrier is robust. Even learning another species song didn't lead to hybridization. But when the size difference between species is smaller, as it is between Scandinavians and Fortis, then learning another species song did lead to hybridization. But very rarely, this is very, very rare, about 1% of all breeding. Now, for the first 10 years of our study, we followed these few hybrids that were produced, and none of them survived the dry season to get even into the breeding season the next year. So we thought, maybe we are seeing the beginning of genetic incompatibility, which would have been very exciting, that we could have been able to follow. But then we thought maybe it's not that, and maybe it's just that a lot of birds are dying now, and there's very little food, and it just by chance these birds are also dying. So it was not until after this El Nino of 1983, when the island was converted from a large hard seed producer to a soft seed producer, that we were able to really test this. Now in 83, this is the survival curve of Fortis and Scandinavians, that parental species. Again, we had a lot of rain in 87 and 91, and these are the survival curve of the parental species. So how did the hybrids who were born at the same time survive? And the answer is, to our astonishment, they were surviving extremely well as, if not better, than the pure species even. So in an altered environment, in a different ecological condition with plenty of food, we had no evidence of genetic incompatibility. They not only survived well, when they bred, they produced as many eggs, as many fledglings, and as many recruits into the next generation. So how did they breed? Well, they followed this rule that they breed according to the song they learn early in life. So the hybrids backcrossed according to the song that they had learned early in life. And introgression occurred actually mainly from the female Fortis into male Scandens. And so what happened from then on, this happened about this point here, what happened from then on, there was a trickle of genes, of Fortis genes going into Scandens, and Scandens' beak shape that started out very pointed became blunter and blunter and blunter and more Fortis-like. So why was this? Well, it wasn't that it didn't go both ways. We did have a very few trickling back between Scandens to Fortis, but it was because the Fortis Scandens female hybrids backcrossed to Scandens males. And the reason was that for this time, for these next 20, 30 years, the sex ratio in Scandens was skewed in favor of males. So it even got to the point when there were about 100 males to only 16 females in some years. So the Scandin males were desperate for females. And being larger than the hybrid Fortis Scandens males, they outcompeted them for territories and for females. So we got the female hybrids, Fortis Scandens hybrids, back crossing into Scandens males. So at this point, we only had microsatellite loci, but there was enough difference to be able to show that the difference between Fortis and Scandens genetically was decreasing, and they were becoming very similar to each other. So the population was becoming very similar and much more different from Scandens on other islands. It was becoming smaller and blunter and more Fortis-like. And this followed our exact measurements over the years this time. But we were wondering if there were specific alleles that could have contributed to changes in beak shape. So once again, we sent our blood samples over to Lake Anderson's lab, and once again, Sangeet did most of the work. So this time, what they found by comparing blunt and pointed beak birds, they came across this Alx-1 gene, which encodes a transcription factor. Once again, this is a gene that is known from humans, and it's known to be very important in craniofacial development in humans, and actually a mutation in the Alx-1 gene is largely responsible for cleft palate in humans. In the finches, it comes in two forms. There's what they call Alx-1 blunt, Alx-1B, that was homozygous in the very blunt beak birds like Magnorostris, and then there was a Conirostris on Hispaniola. And then in the pointed beak species, the Alx-1P was homozygous. In Fortis on Daphne, we got both. Now there'd been hybridization, so we expected both. So, and I remind you that then we got able to show that it was the introgression of the Alx-1B into the Fortis. From Fortis Alx-1B into Scandens, which had been predominantly Alx-1P pointed. So then what our colleagues in Uppsala did was they did a neighbor joining phylogenetic tree on the SNPs that they had got from the genome, and they found, just as we expected, followed exactly what we would expect from our field studies, was that there was a major shift towards Fortis in the autosomes and a major shift towards Fortis in the mitochondria DNA, because remember, it's a Fortis Scandens females that get across. And then we looked at the sex chromosomes, and this also was what we expected from our field data, and the Z chromosome got left behind. Now, in birds and butterflies, they have a different system from humans. It's not the XY system, it's a ZWZZ system, with females being the heterogametic sex. Now, whenever this has been seen before in birds and butterflies, people have said, and many publications that show, aha, this is the reason, this shows that there must be genetic incompatibility because of the large Z effect and the fact that the Z would be incompatible with the autosomes and the mitochondria. But here we know, so far anyway, that there is no genetic incompatibility, and this can be explained solely by social incompatibility. But the intriguing thing now is, could this genetic incompatibility be actually start with social incompatibility and then move on to genetic incompatibility? So that this is what we want to examine much more closely, and exactly what genes are on the Z chromosome. So we had written a mosaic genome arising from hybridization between two species generates phenotypic novelties that could be under selection in a new environment. And this increased genetic and phenotypic variation could possibly fuel a rapid change along a different trajectory towards a new species. And we never, ever thought this would happen. But amazingly, it did. And it happened in the formation of this new lineage of finches. So what actually happened was that a bird in 1981 arrived on Daphne. It looked exactly like, it was this bird here. It looked very much like a tortoise, but it looked like a blown up tortoise. It looked like a giant tortoise. Tortoise is around 17 grams. This was caught by Trevor Price, was on the island. He was a graduate student at this time, studying the finches. He caught this bird, banded it 5110, and he measured it at, weighed it at 28.5 grams. The next year, it was now an adult, and it began to sing, and it had a completely different song that we'd never heard on Daphne, and in fact never heard anywhere on the islands. We came back and we took a small drop of blood from him a couple of years, or I think the next year, and so we had blood samples from this bird as well. So it was analyzing these blood samples later that Sangeet was able to show that this was a member, as we had suspected, it was a member of Conirostris from Espanola. We thought it looked like a Conirostris, but we thought it was impossible for one to get from Espanola all the way to Daphne, but somehow it had done this. And it certainly was a Conirostris from Espanola. It took ages to breed. It was a big bird that got a beautiful territory. It was sitting in this beautiful territory, defending this beautiful territory, but it couldn't get a mate. After all, it had all the wrong song, it had all the wrong shape, and nobody was interested until the very end of the second year of trying. And then a female tortoise mate had died in the last brood of the season, and she went into his beautiful territory, mated with him, and produced a few offspring. And the next year, another tortoise female did the same. And they produced offspring, which we followed, we measured them, we took the blood, and all of them showed genetic transmission from 5110. And all males sang 5110's peculiar song. So then what happened was there was this 2.5-year drought, and 90% of all birds on the island were dying. All these birds died except for these two, an inbred brother and sister. When the rains came back 2.5 years later, the brother and sister bred with each other. They produced 26 offspring, all but 9 survived. So we had a daughter breeding with father, a son breeding with his mother, and the rest of the sibs breeding with each other to produce more offspring which bred with each other, to produce more offspring which bred with each other. All the birds showed genetic transmission from 5110. All males sang 5110's song, and all were large like 5110. So the inbreeding wide coefficient, which is just a measure of heterozygosity across the genome, went steadily up across the generations, as we would expect. But what we didn't expect was that the big bird population, which is here in blue, it was intermediate in body size between Fortis and Conirostris, but it was displaced in that it had a large head and a large bill relative to its body size. So it also fed largely on these tribulus seeds, and it was able to crack them. So we decided to measure the efficiency. And it turned out that it was just as efficient at cracking these tribulus seeds as Magnerostris. And so this is a big bird, so it was just as efficient. And by now, Fortis was so small that it could hardly crack the tribulus at all. So if there was another drought and tribulus was left behind, the big birds would do quite well in competition with Magnerostris because being smaller, they would require less food. However, the new bird lineage is behaving like a separate species. It's much larger than its nearest relative, Fortis. In morphological space, it lies between Magnerostris and Fortis here, and this is the gap that expanded when Magnerostris outcompeted the largest Fortis, which was lying here in the character displacement event, and the big birds moved right in. It has, this is 3 generations of the big bird song, very different from Fortis song, Scandin song, and Magnerostris song. It stays in actually one half of the island. The blue dots are the territories before the drought. The red dots are the territories after the drought. They hold contiguous territories which they defend against each other. But these territories overlap the territories of Fortis, Scandins, and Magnerostris, who ignore them and who are ignored by them. So in all respects, this new lineage is functioning as a separate species. Will it die out through inbreeding depression? It might, but 2 years ago, there were still members of the population there. Will the genetic variation be augmented through gene exchange? It might, but no sign yet. Whether it survives or not, this new lineage gives insight into how a new species could arise and either persist or become extinct. So when we first arrived on the island, there were only 2 species on the island, very clearly separate from each other. After the El Nino of 1983 that completely altered the ecological conditions, hybrids between them began to survive and back cross, and the 2 populations began to fuse. Magnerostris moved in at this time, outcompeted the largest Fortis, and the big birds moved in here. So we've shown insight into 3 models of speciation. Certainly the allopatric model that Darwin thought about. There's also the possibility of fusion between Fortis and Scandins, producing something that is very different from Scandins elsewhere on the archipelago. But then very excitingly, there's this introgression, this hybrid speciation. So how widespread is this hybrid speciation in being a source of variation for adaptation to new environments? It used to be thought, when we first started publishing this, it used to be thought that this was not really important. It just petered out. But now we know that it is very important. It's important, of course, in bacteria, with horizontal gene transfer. It's important with plants. Lauren Wiesberg has done beautiful work on the sunflower, Helianthus, and Richard Abbott on Senecio. It's also now been shown in every single group genera of animals. It's also important, has been important in our own ancestors. There's introgression between Denisovans and Neanderthals and our own modern human lineage, shown by Svante Pavlov. It's also important in cancer clone evolution. We have been asked to go to 2 cancer places, one in Texas, one in London, because cancer clone in, let's say, the colon and the lung is actually taking place within a tissue ecosystem, very much as Darwin's finches were in different islands with different ecosystems. And we also know that there's introgression. The tumor suppressor APC gene function was destroyed due to rearrangements associated with an introgressed retrotransposon through horizontal gene transfer. We also, just last year, showed that there was a genomic region associated with protection against severe COVID-19 is actually inherited from Neanderthals. And this was work that was done by Seberg and Pablo. So we thank our many graduates, postdoctoral fellows, our collaborators, which we couldn't have done this work without, and also Galapagos National Park and Charles Darwin Research Station for logistical resource and our funding agency. But anyone who's worked on this with us will want to leave you with two messages. One is that we live in exciting times. Genomic data is rapidly accumulating. This enhances field studies enormously, but reciprocally, a reliable interpretation of genetic data requires an understanding of ecology, evolution, and behavior, and this is often forgotten by funding agencies. There's one other message, the last one, but the biodiversity is being degraded at an unprecedented rate. We've shown you that the environment changing and constantly changing, and to maintain biodiversity and the potential for future speciation, we absolutely must keep both environments and populations capable of further natural change. Thank you.
Thank you very much, Professor Grant. This last 1 hour has been really, really a life-changing experience for us. And I now request my co-panelist, Dr. Sudhir, to kindly summarize the session for today and also take up the questions which are being shared in the chat section.
Right, thank you very much for that. And so do I go back and let's see, let me click on that. Can I, stop share, okay. Okay.
Yeah. Go ahead, Sudhir. Yeah.
Oh my goodness, 41 in the chat. 41 questions. Would you like to?
No, Sudhir will moderate it for you. There are plenty of them. That's why I've asked him to moderate the things. Yeah, please.
Thank you so much, Professor Grant, for this wonderful talk and sharing with us the insights about evolution in finches, particularly at Daphne Island in Galapagos. And it was indeed a pleasure listening to you directly from you. So thank you so much for that. There are a couple of questions in the chat box, and most of them, since participants are coming from both faculty and students, so there are some generic questions as well. So I'll just take up the questions, and if you permit, and then we can discuss that. First question is from Khushbu Singhal, and she's asking, can birds at Daphne Island not fly to nearby islands during drought conditions?
Yes, I know. This is, you know what happens during drought conditions? They get weaker and weaker and weaker. And what we're doing is when we're on the islands, we just see them becoming very weak, and the first thing that seems to go is that they can hardly fly at all. Now, it might be that a few do fly across, but every day we are picking up dead birds, and of course they're banded, so we know who dies and who survives. So all we can do is to look at the vast number of birds that we see who have died and who have survived, and just assume that there hasn't been one or two that have gone to other islands. Whenever we've been on other islands and do see birds come across, they come across at the end of El Nino year, when the populations have exploded, and then they seem to disperse. But there seems to be, or we have never seen, doesn't mean that they don't, but we've never seen a dispersal of a bird in very weak condition.
Yeah, thank you so much. There's another question by Nitin Sahi, and he's asking, does Darwin's theory matches with ancient gene theory?
How does Darwin's theory match with the ancient
gene theory? Yeah.
Sorry, what was the last word?
Gene theory.
Gene theory. Okay. Yes. So Darwin thought that actually we would never see it happening in contemporary time, that it took such a long time, took millions of years or thousands of generations before we would actually see any difference. But the idea now is that these things are very small differences that occur in tiny increments, which eventually accumulate till you get much larger differences between species. And you can see this sort of thing happening a bit in the fossil record, where you get this, and you often get, stasis where there's not much change. And then sometimes you can get a sudden change. And often this sudden change is associated with a strong ecological event of some sort or other. So we are just seeing, with the drought, we're seeing really just a small increment. But if you think of a really large change, then you're likely to get a much bigger change.
Right. So another question is from Ashish Russell, and he's asking how hybrid birds are not sterile. Hybrid bird offsprings are not sterile.
Well, eventually they will become, for reasons of genetic incompatibility, but this can take quite a long time. So we have a period when there are very, very few hybrids being formed, and occasionally they will be able to backcross and form viable offspring. But this is a gradual change. So for example, I said that in pheasants, it can take something like 37 million years before they have genetic incompatibility. But there are other species that become genetically incompatible in a much shorter time, some 5 million years, and then or less. And then some can become genetically incompatible sort of almost immediately if there's been a change in genotypes, particularly in the sex chromosomes. So there's a variation of time depending upon the genetic change. So,
yeah, thank you, Professor Grant. Sambita Das wants to know, is there any relationship between evolutionary timing and the complexity? Can we say more complex organisms take more timing compared to less complex one, or it depends simply upon the efficiency of mutational events?
Sorry, I didn't. I'm going to. So I realized that something's just come on the phone. I'm just going to stop that so I can hear your question. I'm so sorry about that. Okay, could you repeat the question?
Yeah. So Sambita is asking, is there any relationship between evolutionary timing and the complexity? Can we say more complex organisms take more timing compared to less complex one, or it simply depends upon the efficiency of mutational events?
Yes.
So basically, basically she's asking if there is any relation between the time evolution takes with respect to complexity of the organisms.
And so you're thinking that it might take longer. Okay, there's a real difference between the time that it takes between mammals, and this is very interesting because in mammals it can be much faster. And the thing about mammals is that you can get it can take much longer in birds than in mammals. And in mammals, you have the placenta. And it's thought that there can be genetic incompatibility occurring very fast in mammals because of the differences in the immunological conditions in the placenta. So yes, you're absolutely right. There's a difference in the time it takes to becoming genetically incompatible.
Right. Shivani is curious to know, what could be the leading cause of social incompatibility? Isn't it related to genetic one in turn?
Oh, so in the social incompatibility with the tortoise and scandens that we saw, this was due to, well, it is related to genetic incompatibility because in an indirect way, because of size. It is the scandens, size matters and size is dominant. And so because the scandens males were larger than the hybrid males, then they outcompeted them for territories and females. So in this way, yes, the size, which is genetically largely determined genetically, mattered. So it matters indirectly like that. Song is learnt, but on the other hand, the ability to learn song, just as the ability to learn speech in humans, is genetically determined. So for us humans or the birds, the ability to learn is partially genetically determined, but what we learn, what language we learn, and what the song the birds learn is, of course, culturally determined. So there is no real clear distinction like that. You know, there's always a genetic underpinning to something.
Absolutely, absolutely. So Dr. Prasad wants to know the gene described by you, which is involved in speciation, codes for which protein?
Oh, I have got to go back and see this because I can't do this. All I can say is off the top of my head that they are transcription factors and I can't tell you off my head. I can't remember.
That's okay. We can.
It codes for, but actually it is in the papers, and it's the papers where Sangeet Lakmani is the senior author. A large number of authors comes out of Leif Anderson's lab, and it is in the paper there and in the supplements of those paper. And I will probably tell you the wrong thing if I try and do it off the top of my head. Sorry about that.
That's okay. That's okay. So Dilawar Singh wants to know which is fast, genetics or epigenetic changes, since both are, affected by perturbations due to environment and other factors.
Right, and the epigenetic changes obviously occurring, and we haven't looked into those yet, except that we are interested in doing so. So there are epigenetic effects that must be going on. I mean, we've seen these effects on these big transcription factors happening within a generation. So that's fast. And if there were epigenetic changes, that would also have to pass down the generations. You actually have to check it to go through from one generation to the next. So we know that the two that we have looked at, we see changes from one generation to the next. And epigenetic changes, the way we would look at it, we would also look at it from one generation to the next.
Right. Professor M.V. Rajam, he's actually giving a comment that it was indeed a very excellent talk, and followed by which he is a question. What is your opinion on Darwin's and Lamarck's view of evolution in the present scenario where epigenetics plays a pivotal role in organism development and evolution? So it's just a related question to epigenetics again.
I know, actually, you would probably be interested to know that when I first started as an undergraduate, I did a special course with Waddington and in Edinburgh, and of course the Lamarckian. This was, very much a discussion that was going on when in the 1950s, late 1950s, when I was a student with Waddington. So the there are connections. I mean, it depends when you go back to Lamarck's work, how you sort of interpret the Lamarckian part. But if you think of Waddington's heat shock proteins and this sort of thing, so changes can actually happen in a sort of, as some people would say, Lamarckian type of way. That's a rather roundabout answer to your question. I don't know if you want to follow it up with something.
Yeah, no, the questions are pouring in because of the wonderful topic and your wonderful talk. But I think considering time, the last question that we can take is by Mostushi, and she's asking what type of speciation found in plants in those islands, if you want to comment upon that.
Oh, the types of speciation found in plants. I mean, this is really interesting because this follows almost exactly this beautiful work that has been done by Lauren Riesberg, and which I referred to on the sunflowers. And he has shown exactly the same sorts of things that we have shown, particularly with introgression, which has introgressed certain alleles. And this has allowed the sunflower helianthus that he has worked on to actually to invade places that, like very salty places, very dry places, or very cold places. So this follows almost exactly a parallel to what we have been showing as well. So it's very exciting to see this parallel. And I should maybe add that you might be also interested to know there's the same sort of parallel happening in butterflies, where it's particularly the butterflies that mimic each other. So there is, even though they've been separated for large numbers of generations, there's the occasional gene flow that goes across, and this takes the patterning alleles with them that then is selected for in areas where there is a common enemy, if you like a bird that preys on them. And so this is a very good explanation now for why you do get these mimicking rings that occur across different species. And it's exactly the same. It's exactly the occasional hybridization where a gene goes across that is then selected on in a different environment. So that is extremely exciting. Again, exactly parallel.
Thank you, Professor Grant, for addressing the queries so patiently, and it was indeed a very enriching experience for all of us. So thank you so much for that. Over to you, Professor Manoj.
Yeah, thank you,
Thank you very much.
Thank you very much, Professor Grant. And it's around 8:40. It's a dinner time over here, but I must appreciate that there are still more than 120 people who are still there, and they've been listening to you, and they have been continuously asking me that when we are going to upload this this talk, the recording of this talk on the YouTube. So I assure all of you that we'll be uploading it very soon. And on behalf of the National Academy of Sciences India Delhi chapter and the host institution, the Deen Upadhyaya College, I would like to thank you once again for sparing your valuable time and being there with us and enlightening all the students and faculty members.
Well, thank you very much. And I wish I was in India with you.
Sure. Thank you
very much indeed.
Okay. Okay, fine. Thank you. Bye-bye.
Bye-bye.